my name is Toby Gibson, and I started at EMBL in 1986. I came on a 2 years postdoc, and now I'm actually retiring from that 2 years postdoc at EMBL.
Fantastic. That's been one of the most productive postdoc periods that I think we've heard of at the EMBL. We started out, I think, on the same day as PhD students in Cambridge. That was a long time ago now. You were a PhD student with Bart Burrell. What was that like as an experience?
Correct. Yeah, I enjoyed it, obviously, but what I did was, let me just even start from earlier. I studied molecular biology in Edinburgh, and then I went to Cambridge to do my PhD, and Bart Burrell was part of the Fred Sanger's DNA sequencing outfit department, and had worked on developing the methods and working on larger and larger sequencing. So in those days this was fully experimental, bench-based, running sequencing jails. And we collectively sequenced Epstein-Barr virus. It was about 120,000 bases, and I did 20,000 bases. And then obviously running just through sequencing is routine work. So my sort of interest in when I was thinking about things would be how can we improve the methods? And one thing that I was able to introduce was using sulfur-35 labeled ATP in the sequencing jails. And this was useful in a couple of ways, S35 being an alpha emitter compared to P32, which is a beta emitter, made much sharper exposures on X-rays because the alpha particles don't penetrate very far. The other thing is that the half-life was longer, so you could use the material for longer, that you want, the radioactive material. One other thing I did then was also to engineer, do a little bit of bacterial strain engineering. The strain that we were using, which actually was developed in Heidelberg by Joachim Messing, was supposed to not have any restriction site activity, but it became clear that it did, not particularly strong, but it was there, and that was hampering our efforts to complete our sequences. So I was supplied with a phage from Norrin Murray in Edinburgh, who had been a lecturer of mine, and I was able to remove that site using her engineering tool. And then I called it TG1.
Yeah, I think for young people today, scientists today, the art of sequencing DNA has become a fully automated, outsourced activity. I think for many of them, they won't even understand what you're talking about. Yeah. When you describe it, I mean, it brings back a lot of memories to me because, as you know, you not only innovated in developing the technology, you were kind enough to teach me the art of DNA sequencing when we were both PhD students together. Might be helpful just to remind people how DNA was sequenced in those days.
Yes, so you needed to have cloned material in plasmids in bacteria, and so when we sequenced Epstein-Barr virus, we were supplying clones by a group that had completely overlapping sets of clones for the whole circular genome. Then we would shotgun those, so we would actually sonicate the DNA and then purify out sort of 300 to 600 base sections and reclone them in our M13 vector, which was for making single-stranded DNA for the sequencing. We then run the sequencing reaction, where you had to use chain terminators for tcGNA and the radioactive label, and then we would run these out on quite long gels, and then we would have to get them in the computer. We were already generating quite a lot of data for wet lab people, and so we need to get them in, and we used digitizing tablets, and we would go up with a pen and record all of the bands. So then the main problem with the route that I had taken is that I knew how to sequence DNA, but I didn't really know much else, and it's much more, you go into science for various reasons, but a lot of people would like to be able to do the beautiful experiment. But I was just sequencing DNA and working on the methods.
Well, I mean, it was a beautiful method.
Yes.
There was a Nobel Prize awarded
there is
for
beauty in there, but,
yeah.
but I wondered if I might scale up to a bacterial genome, and I spent 2 more years in Cambridge trying to make the clones for that, but I ran into an artifact where I failed to cover the genome. I don't really need to go into, but I did some, I was able to publish some more plasmid cosmid engineering and stuff, so I got something out of it, but I didn't.
That was when you worked with John Sulston. So
yes, I was.
You did the experience of working with 2 Nobel Prize winners.
Yes, so I think I was kind of officially with Sidney Brenner, but he was head of the institute by then, and I only talked to him a couple of times. So effectively, I was with.
He was also a Nobel Prize, so that was 3 Nobel Prize winners.
He wasn't then, though.
You were trained by, yeah.
Anyway, yeah. So then I was sort of, on this track where I couldn't do the beautiful experiment. I hadn't prepared my library that I was going to start sequencing. Also, I was in a kind of limbo situation because there's not that many places were doing sequencing then, and I would probably not get hired anywhere, and also wouldn't have the funding to do it in a lot of places. So I didn't know what to do. Then I had one of those sort of sleepless nights, where your mind wanders, and suddenly I realized what I needed to do was just go on the computer, because the sequences were going to come there. And so I still thought that was a good idea in the morning, and continued to think that. So yes, I thought I'll go on the computer, because the data will come. And then the place at that point where it was logical for me to come was actually EMBL. EMBL already had the data library. It was well established. It had computers, which obviously were necessary, but that meant I could use them. And it had a vigorous structural biology program, also heavy computer users. And then it turned out that it was going to set up, and was in the process of setting up, a bioinformatics unit. And I happened to come to a workshop, so I talked to people who were likely to go into that unit, including Pat Argos, and then I applied to the Royal Society, SERC, NATO combined European Fellowship, which was a very simple form, which made me think that so few British people were coming to Europe, and that they didn't need to really query us very much.
And what was EMBL like when you arrived? Because obviously today it has grown enormously. We're sitting in the wonderful ATC Centre. It's a much larger institute. Yeah. Just give us a picture of what it was like when you arrived. Well, it was
obviously smaller, and it was still a very young institute, although I should say that the institute was older than my scientific career, so I didn't particularly think it was a young institute when I came. Obviously when an institute is set up de novo, there is a kind of a risk if you come and work there. So I think some of the early sort of group leaders were really, there were some characters and some people who were not intimidated by the fact that this was slightly risky to come. So after the 2 years, I actually was able to stay on working with, in the structural department. There were people who worked on muscle proteins. We had a visitor, John Trinick, from the UK, who came and increased the activities there. Kevin Leonard was very active, and others. So we ended up actually looking at quite a lot of muscle proteins. These are not the ones I'd first been looking at, but this was what happened. And I ended up then actually being in Kevin Leonard's group for a while. He's an experimentalist, but we were just working on with sequences and looking at structures and so on. And then increasingly collaborating with people like Annalisa Pastori for NMR structure determination and so on.
And these were the very early days of the origins of the field of bioinformatics. And back then, if I remember correctly, the EMBL had the data library, which started off the process of gathering together and making publicly accessible all the sequences that were now being generated in copious amounts by different groups around the world. Absolutely. And so, the data library, I think, was a great initiative that Graham Cameron set up. And
I mean, it laid ultimately the groundwork for all of the bioinformatics services and support that EMBL provides, most of it, but not all of it, in the EBI near Cambridge. And then a few years later, my collaborator Des Higgins came, and he was in the data library, though not really purely doing data library work. He was he had already developed software to do multiple sequence alignment, and I still used to do quite a lot of my alignments by hand when he came, and so we would joke about that. But we eventually got together, and at a certain point, I also had become a staff scientist, and I had one person working with me. That was Julie Thompson, who was a mathematician and C programmer, and Des and I and her, we discussed various new ideas for the next version of the Clustal software, and she implemented many of those ideas and tried to explain to me when some of my ideas were unmanageable and could not be converted into code and so on. But the end result was that in 1994, we published the Clustal W version of the series of alignment software. And this caught a wave of sequencing. There was more and more sequencing coming, DNA sequences, and then if you're interested in the proteins, as we primarily were, the translations also go into the databases. And we were lucky with the timing because it became massively successful. Des very keen that the software was available for everybody to use, open source software. He was very keen that it ran stably, and so we used ANSI standard C. So in principle, it would compile on any platform, PCs, Macs, but in those days we would use it on Unix computers.
So these are obviously the hallmarks of making great software tools, the stability and efficiency and easy use. I mean, that was a fantastic time, I guess, to be in bioinformatics when for the first time ever, large numbers of sequences were being generated. There was clearly a need for these tools. I guess today many researchers know about the tools that are available, don't remember or never had experience where they came from or what it was like. Tell us a bit more about Clustal. I mean, what is Clustal? How did you come to develop it, and what do people use it for?
Right. So so Des originally developed the first Clustal when he was a PhD student in Dublin, and their group was working on HIV, evolution of retroviruses and pathogens, and they needed alignment software. This was back in the 80s, so this was slow computers. And he though there had been previous work, he was able to create software that essentially combined steps which people hitherto have been running individually, one after the other, and he could put them all into one program. And because the computer is so slow, you would not have very many sequences, and it would sort of run all night and stuff. And he wrote a Fortran program. He updated that a few times.
And this would compare different sequences that people had generated?
Yeah, so for example, if you were interested in HIV, well, there's other retroviruses, and therefore if you wanted to see, for example, you might want to line up some HIV proteins with other retroviruses, and there's various reasons for doing this, but one of them is the amino acids in the proteins that are conserved are either structurally important or functionally important, and therefore I'm still today using alignments all the time to try to ascertain where function might be in protein sequences. We often nowadays have structures. We might have had structures in those days, and then the function. A lot of the function is on the surface of the proteins. Most proteins, maybe some of them have internal function too, and therefore you're interested in the conservation on the surface. The conserved patch on the surface is doing something. So if you don't know what it's doing, you have a target to work on. There are many other purposes. I mean, the evolutionists are just making alignments in order to study evolution. This is still a major activity today. And if you think it's still in the viral theme, you're looking at things like how coronavirus evolution and where SARS-CoV-2 has come from and so on. All really important things that you have to do. So lining up.
Sequence alignment is really fundamental to being able to turn sequence data into knowledge.
Yeah, it's fundamental to modern, what I would say, biomedical science, which is the era we're in now. I was a molecular biologist. Yes, and we caught that wave of sequencing, and so this is rather nice for my career, and I guess the three of our careers, because we had provided this tool that was so widely used and so highly cited, it became EMBL's top citation, and
that's
I sort of felt a little bit secure in my career, shall we say.
Remarkable achievement. Can you give us some concept of just how many people have used this software that you developed?
Well, the citations, of course, doesn't. I mean, tens of thousands of people, hundreds of thousands, possibly. I really wouldn't know. I mean, it depends how many scientists there are really in biology who need to make alignments. Bioinformatics tools don't get cited in every paper they're used, so yes. they get cited when it is necessary. So if you need to show an alignment, for example, you should say where it was made and things. Yeah, so that was nice, and we continued looking at protein domains. It had become clear in the 1990s that proteins, as they were first understood, like the structures of myoglobin and hemoglobin that were solved in the LMB in Cambridge before our time, were single-folded units. It became clear that lots of proteins were much bigger, and that they weren't single-folded units, and that they could have multiple domains. And so we would be using sequences of these proteins to identify regions which might map to other proteins which only had a part of parts were similar, other parts were different, and so on. And there became a big activity in identifying these domains. I'd particularly single out Pierre Bork, who's still here as well, for finding many more than I did, but we found some number, and so this was sort of era in the 1990s. But we had this problem that there were regions between these folded domains, which rapidly, once they had been identified, people would tend within a couple of years to have solved structures of them. So we'd have alignments and we'd have structures, and hopefully function would come. But there are these regions that clearly couldn't fold because they didn't have the hydrophobic amino acids which you have in the core of a folded protein. And these could be very big, and this had come out of the sequencing of genome efforts. And then people started finding very short little bits of proteins that we call sequence motifs. And the reason we say it is a motif is because it comes out of a sequence alignment. We're looking and we see a motif down this set of sequences. And those were slowly accumulating in the 1990s. And but actually, I knew about them from our PhD time, because people like Colin Dingwall and Ron Lasky were working on the nuclear localization signal, which was a small positively charged motif. And we knew them, and we saw them do presentations. And then Tim Hunt, who wasn't at the LMB but was uptown, and who discovered cyclins, it turned out that cyclins bind to a motif, and that the cyclin-dependent kinase, protein kinase that they interact with, would then do a modification called phosphorylation on substrate proteins which bound to the cyclin. So the cyclin would direct these things, and so you'd have the two motifs. And then other motifs appeared in cell cycle, because these are cell cycle stories, such as the D-box or destruction box, which was in several of these cyclins and caused them to be rapidly removed when part of the cell cycle that they should go through had been finished. Of course, you know all this because you've worked on cell cycle, but
I know a little bit about it. I mean, it's clear you've helped to develop the language of proteins and the alphabet of proteins that you make up structures and functions. How do you look on this now? I mean, after this long, successful career, seeing the field start from its very initial origins as the first sequences could be determined to this stage, do you see that as a solved problem, or what are the exciting frontiers in sequence alignment and motif detection today?
No, it's not at all a solved problem. So the something that I used to get frustrated about, because obviously by looking at a lot of different proteins, you start to sort of see aspects about how they might be working, and you start to realize that there is a lot of modification of proteins regulating their function. And so, for example, when phosphoproteomics became a thing, which is a method to pull out the phosphorylation sites and could be applied at relatively high throughput, you quite rapidly got to like 100,000 phosphorylation sites in the human proteome, 100,000 in the mouse proteome, which we don't know what they're doing. And we still only know a rounding error of what's going on there. So there's more and more evidence that complexity in cell regulation and cell signaling was badly underestimated, and there will be a lot of over simple models of this. It is getting better now. In fact, it turns out that one of the various things happened. The mass spectrometry people who are doing proteomics, they would look and see if a protein was interacting with other proteins in the cell. And pretty much every protein that they would pull down as the tag would bring other proteins down with it. So it became clear that proteins are almost always in complexes with other proteins. And then why would you therefore have a model in which your protein kinase is diffusing across the cytosol, which anyway is full of blockages from vesicles and things, and its substrates are diffusing, and you have this sort of billiard ball model of the things interacting and stuff, and this couldn't be the case because they pulled all these proteins down. And then additionally, resources like the Human Protein Atlas, which has tried to make antibodies to all human proteins and then see where they are in a cell. Almost every protein that you look at has a spotty pattern. So it's in discrete localizations, it's not uniform. You could find some uniform ones, but the great majority of them are highly discretely localized. So once you see that, then again, you can't just say things are diffusing around because they're in spots.
Well, I would argue that their distribution isn't static for many proteins. So when you say where are they, it depends at which time point, because that can change.
Oh yes, no, things are highly dynamic, but you just get a snapshot when you just label the cell. But there would be a model that there would be like there's a famous cascade, and I don't like cascades as a metaphor, except in limited circumstances, but the MAP kinase cascade, and so you will just see pictures in the literature where there's a perturbation at the membrane, so some hormone binds. It then leads to MAP kinase, phosphorylate, MAP kinase, phosphorylate, MAP kinase, phosphorylate, MAP kinase, and then it leads to transcription, so gene expression, genes being, proteins, RNAs being made in genes, active and making proteins, and that's always just shown as this linear thing. It doesn't work like that. And the information also goes through multiple ways. Information from the cell surface doesn't only go through one way, it goes in other ways, and that allows communication, networking, and therefore the information content of different assessments that the cell is having to make all the time. I mean, you could be sick because you've got coronavirus, you could be starving, it could be 40 degrees. Your body has to deal with all of these things and somehow try to keep you alive. And, whereas we're just sort of sitting here, and I guess we had some breakfast today and maybe some lunch. And so most of what's going on in most of our cells is just ticking over very quietly and peacefully, hopefully. But another of the paradigm shifts was when people realized that there are actually liquid complexes. So instead of everything being neatly stuck together, there's what's called liquid phase phase separation. And many of these complexes have a component of RNA in them, but they don't have to. And they have proteins which have these large unstructured parts which can be modified. And therefore, you can change probably the liquid-liquid phase separation by modifying many sites on individual proteins. And this is a completely different way of understanding how you could regulate systems, because we always used to think, well, if there's one phosphorylation site, it has one function, and it might be to bind another protein, or it might be to block the binding of another protein. So this would be sort of one-on-one type interactions. But there's proteins like SRRM2, which you will know the name of, and it's sort of got nearly 2,000 amino acid residues of disorder, and it has hundreds and hundreds of phosphorylations whenever somebody does phosphoproteomics. And I don't think anybody knows what that does properly yet, but it's presumably a phase separator under certain conditions involved in splicing.
I think, it's a subject dear to my own heart.
Yeah.
You've touched on a number of really important topics. I think they're good to unpack a bit more. I mean, one, life is complex. As methods develop, we find ways of measuring things we couldn't measure before, and you highlighted some things we still can't measure comprehensively, so we don't have clear oversight or insight into functional relevance. I was struck also by one of your earlier comments, Toby, how in the early days of developing Clustal, the computers you had were very slow by comparison with modern computers, and that limited again what you could do. Well, I mean, computer science and computer hardware is another field that has just seen explosive growth during the time course of both our careers.
Yeah.
And I think that was really brought home a couple of years ago with the impact of AlphaFold and the whole idea that neural networks and machine learning, AI, using all these different terms about approaches beyond sort of more conventional top-down programming approaches could achieve results that were, remarkable.
Yeah.
I mean, what are your thoughts about the impact of AlphaFold on your field, or more recently about these new large language models and transformer models and all of the deep learning technology that's now impacting on us?
I'm retiring, and therefore I'm not going to see the full flowering of these technologies. But when AlphaFold first came out, people like Alex Bateman in the EBI were amazed at how good it was. I mean, you have to be careful about saying that these are predictions, because what AlphaFold is using are known structures and huge multi-sequence alignments. And one of the things it's looking for in the multiple sequence alignments is correlated mutations. You need a big alignment for that. They're very weak. But why correlated mutations are desirable is that if you've got a folded protein and then inside you've got these hydrophobic positions, amino acids all neatly packed, if one of them goes from a small to a very large one, it probably degrades, it may even unfold the protein. So they can, over time, incrementally change by a small one getting a bit bigger and a bigger one getting a bit smaller and so on. And so these can be detected, and then they can be applied in predicting protein folds. But in addition to just having that alignment, you have the existing structures. And so it takes all of that stuff. It's a black box, what exactly it does, but you give it now a protein sequence where there isn't a direct structure, and it may be able to work out that what the structures are that it can do. And so people like Alex Bateman in the EMBL got very excited about this, because they could now make candidate domain structures in proteins where the sequences are actually so divergent that the similarity is random, and yet you could now get a fold. Could be wrong, but it has its assessments. So if it's very confident, it probably is right.
And it's testable.
And it's testable. I mean, you can obviously go out and if it's a multi-domain protein and it's given you certain regions, you can try to now solve those structures and so on. But people like Alex Bateman are a minority. As soon as it came out, people wanted interactions, and it was not trained on interactions. So they were sticking two proteins together, actually, because they could only give it one protein, so they stick two proteins together and does it interact, and so on. And it absolutely wasn't trained for this, but this is what everybody was doing in bioinformatics. And this obviously feeds back into DeepMind, so now they make variant training and they try to bring in training on complexes, and so they provided these. Now, the problem that I haven't fully explained what we've been doing the last 20 years, but I haven't really done it at all about the short motifs, but we have a database of these short motifs. We started it in 2001. And it slowly grows over time. And so you can take examples out of our database or just from structural databases. And in principle, you can try look at whether you can train and so on. But also you can just ask, does this protein, because these two proteins interact, does this bit of the protein interact with this bit of motif, a bit of sequence, and so on. You could. Now, because this is very sparse information compared to where you've got whole domains binding whole domains and much bigger interfaces. At the moment, it's not very good. People are playing a lot, but you get something, but it needs to go through more development. Now, you can't predict the future, but I don't see why things just shouldn't get better with more training and so on. And as you mentioned, there's not only one kind of algorithm, so people can try different artificial intelligence approaches. And so.
I think we've seen that already, clear examples. The one version of a neural network technology doesn't look that impressive, and then suddenly it goes from being not that impressive to being, oh my goodness, that's remarkable. So do you see neural networks and AI being the future in this field?
You know, they're part of the future, but there's always rubbish in, rubbish out, right? If you set up things wrongly, you still get nonsense, and you have the problem that if you're dealing with a black box system, it's very hard to work out why it's producing rubbish, other than it hasn't been trained well enough. So you know, in the protein interaction field, we are very excited. But there are many limitations. For example, AlphaFold is not trained on post-transcriptional modifications, of which phosphorylation is only one of 300 different kinds. So it doesn't know about those. Yeah. And so then those kind of interactions can't really be reliably assayed.
So there's still space for experimental science to actually go and generate the data that can be used to train the models.
You want to validate experimentally anyway, even if you use AlphaFold for guidance. You need to get into the lab and not just think everything's there. But you know, there could be other things, like, because we actually know now quite a lot of proximity in networks, you may just know that, you know, certain proteins are together, but you don't know which ones interact. You can now, if you can afford the computational time, because it's costly, but you can try different proteins to see which ones might come together, and then you could follow that up experimentally.
You're coming to the end of your major part so far of your experimental work. Yeah. If you were starting out today in the field as you see it at the moment, what would you decide to work on?
I don't know, because I mean, the difficulty is that why did I become a biologist? Because I'm really a biologist on the computer. I'm not really a computer. I'm not a computer scientist or whatever. And
that's a very interesting point. So, because people approach this from different areas. There are people who come in as mathematicians or computer scientists who then apply their skills to biology. But you're a great example of someone who was an experimental molecular biologist, hands-on, wet lab, bench scientist, who's then gone the opposite direction in silico. Do they tend to work on different projects or think about projects in a different way? I
would say yes, definitely. Yeah, because I'm you know, I guess I was a bit of a naturalist as a kid, and I just sort of progressed going to university studying biology. Realised I could go into research because I didn't have that background in my family, and then ended up on the computer because of the constraints discussed earlier. Interestingly, Des Higgins is the same. He's still bird watching and stuff, and so, you know, biology is fascinating, and it is now absolutely biomedicine, the era. A lot of it is extremely collaborative with different levels of expertise that you have to have. So you know, if you were trying to, let's say, you know, where are new cancer drugs coming from? Well, a drug is always they're almost always inhibitors, and they have a target. So you can get that target experimentally, but then would it work in the body? And there are many, many steps to go. But in the end, if you were working on cancer drugging, you need to work with clinicians, even if you yourself are a wet lab cell biologist or working with cancer cells or whatever. We've had a phase of enormous amounts of DNA sequence. So we have massive amounts of cancer genes, we have massive numbers of different species and so on. I think I wouldn't go into sequencing now. It might turn into, as if I wanted to be a researcher, it might turn into a routine that you would have a good career, because the amount of sequencing will never go away. But you know, it may be that hospitals will start having sequencing setups, cancer hospitals and so on, and just be sequencing away. And so you could be providing valuable service. But I think it's less interesting to be a sequencer right now if you want to do the research. And we noticed, because I've been in protein bioinformatics pretty much since I've been at EMBL primarily, and it became unfashionable. All of the computer scientists were going into DNA and RNA. And actually one of the consequences of AlphaFold is that protein bioinformatics is back now. It's going to attract more people into the field and things, which is a good thing, because most of the function, not all of it, but most of the function is at the protein level. And in the end, we have to get at that.
We're coming to the end of our chat. Just to finish up, I wonder if after this long, successful career, you could tell us what you will best remember about EMBL.
It has, I mean, it's done many things. I'd say it's shown the value of collaboration. When, as I mentioned, when I came, not many British people would come to Europe. They'd always go to Australia or Canada or the US, but, you know, Foggin Channel, Europe cut off and stuff. Now, as part of the connection of European science, it isn't only EMBL that has done that. The EU's obviously worked towards that with its grant funding networks and so on. But EMBL has played an enormous role in networking Europeans, training up young group leaders who then they may go back to the country of their original nationality, or they may go to another place in Europe, they may go somewhere else in the world, doesn't matter. It's all part of this network of people who have learnt the value of this rather open, interactive environment that we have. And, you know, I'm just so lucky that I have spent most of my career here. It's incredible.